Bismillahirrahmanirrahim. Our today topic is about chromosomes. Cell contain nucleus and inside the nucleus chromosomes are present. Chromosome is the genetic material. Inside the chromosome is actually made up of DNA. DNA is a long tube. This long tube short itself by revolving around the histone protein. It is like a thread on the tube, like we use it for sewing. Okay, this is like this. So this is the thread which is revolving around the histone protein, and this thread is made up of DNA which is revolving around the histone proteins, and as a result the DNA becomes short and condensed and acquire the structure of this chromosome. So this DNA is actually made up of genes. Genes are actually responsible for making special type of proteins. Proteins for hair color, proteins for skin, proteins for each and everything, like for the nails, hairs. So each sections contains genes, and this DNA is too long, it's revolving around histone, and after revolving it becomes short in structure. DNA and proteins together form chromatin. DNA and proteins together form chromatin. And some RNA is also the part of the chromatin. Some RNA also present in the chromatin structure. Okay. Now the second is nucleolus. What is nucleolus? You know about it. Inside the nucleus there is a dark stain is present that is called as nucleolus. Function of the nucleolus is to form the ribosomes. It forms ribosomes. Like it has its own DNA. Do remember this, that it has its own DNA. Nucleolus has, this is present inside the nucleus. Nucleolus has own DNA. इसका अपना DNA होता है. These DNA have their own genes. Nucleolus has own DNA. This DNA has own genes which is responsible for making the ribosomal RNA and transfer RNA. So what we said that inside the nucleolus is present. Nucleolus has own genes. Its DNA contains different genes. Then the genes which are present on the chromosomes in the nucleus, that one is not present in this. This DNA has own genes, which genes are responsible for making the ribosomal RNA and transfer RNA. So it means that nucleolus make the ribosomal RNA and transfer RNA. These genes which make the transfer RNA and ribosomal RNA. More ribosomes means larger the nucleolus. Why? Because nucleolus is responsible for making ribosomes. So if the ribosomes are made inside the nucleolus, so it means the larger the nucleolus, the larger will be the more will be the ribosomes. Different parts of nucleolus during manufacture of ribosomes are involved. Different parts of nucleolus are involved in the formation of ribosomes. But during the cell division, as we all know that about in mitosis and meiosis, when one cell is divided into two, and when the cell is dividing, this nucleus membrane and the nucleolus will be disappeared. We discussed about it in the mitosis and meiosis section, that the nucleus and nucleolus will be disappeared. So in cell division, mitosis and meiosis time, ribosome synthesis will be ceases. Why it will be ceases? Because this will be disappear at that time. So this at that time it will be ceases. Now we come to the endoplasmic reticulum. We know these are the flattened compartments. These are the flat compartments having cisterna. Different processes occurs inside in it, which is different from the surrounding of the cytoplasm. Like molecules, it makes special proteins. This makes special proteins which is transported from the endoplasmic reticulum, and it is separate from the other proteins which are present in the cytoplasm. Means the endoplasmic reticulum is making the protein that is different from the protein of the cytoplasm. Now it has two types, rough and smooth endoplasmic reticulum, means which is present on the look at diagram, the endoplasmic reticulum which is present over here. There are two types of endoplasmic reticulum, one having ribosomes are present, and one is just a smooth endoplasmic reticulum which having no outer structure present outside. And look at this is rough structure. This is called as rough endoplasmic reticulum, and it is a smooth structure. So the rough is responsible only for the protein synthesis, Rough is only responsible for the protein synthesis because ribosomes are present over it, so it is rough. Having black dots are present, ribosomes, and smooth is responsible for making lipids, steroids, lipids like cholesterol, and steroids which is responsible and involved in the reproduction process, like hormones, female hormones, male hormones which are involved in reproduction is formed inside the smooth endoplasmic reticulum. This is the main functioning which is performed by the smooth endoplasmic reticulum, and the main thing is that the calcium ions are formed in it. So therefore, smooth endoplasmic reticulum is so common in muscle cells. Smooth endoplasmic reticulum is so common in muscle cells because of these calcium ions. Muscle cells contraction is because of these calcium ions, and this is done by smooth endoplasmic reticulum. The next topic is about ribosomes. Ribosomes are small. It is not visible with the light microscope. High magnification will be used to see it. There are two subunits. One is denoted by, size is denoted by S, Savitberg unit. What is Savitberg? It is the unit which is used to measure to distinguish between the ribosomes. Their measurement help us to distinguish between the ribosomes. For example, if they are high sediment, high matter, it will be moved down. If you are placed in a centrifugation machine, this is one of the machine, and I put the ribosomes in it. If the ribosome, or you can place any material inside in it, centrifugation machine, the sediment which are of high weight, they will be moved down, then the lower weight, and then the more lower weight, means the concentration will be different. So this sedimentation power shows us which ribosome size is larger will be moved down and the lighter will be on up. S units will be, the faster they sediment, the S number will be high, means Savitberg unit will be high. The smaller later they sedimented, means the S number will be low, means the Savitberg unit will be low. We are discussing in terms of unit. If the unit will be higher, it means it will be more smaller in seconds. No? It's smaller in size too. And if it will be taking too much time in sedimentation, it means S number will be high. It will take more time. Come to the prokaryotes, they are 70S, eukaryotes 80S units, S units, Savitberg units. They are larger in size, they are smaller in size. Formation is done by ribosomal RNA and proteins. Ribosomes are made up of ribosomal RNA and proteins. These main function is allow in interaction of the molecules. Ribosome help us that different molecules will interact and make the proteins. And you know about the transcription, that how the messenger RNA, how nucleus send a messenger RNA down in the cytoplasm. In the cytoplasm, how transfer RNA brings amino acids, and this messenger RNA, comes from the nucleus, and ribosomal RNA is reading the things. Ribosomes are reading even the correct matching is done or not. So this is the process how the ribosomes are interact the things. Ribosomes interact. Transfer RNA is taking it, the amino acid, messenger RNA help in combination. So these all things are involved in protein synthesis, which we discussed in prolonged inshallah in transcription process. So this is the ribosomes actually help in interaction of the molecules in protein synthesis. And do like my channel. Thank you so much. अगर आपको मेरी वीडियोस अच्छी लगी हो तो इनको सब्सक्राइब करें, लाइक करें और बेल आइकन को प्रेस करें. थैंक यू सो मच.